Welcome to Biology 2402 Lecture Series, Chapter 20, The Heart. Chapter 20, The Heart, is an extension of Chapter 19 on blood, and it also relates to Chapter 21 on blood vessels. We're going to start with this first slide on pulmonary circuit and systemic circuit. Do make a note of this slide. Now, for me, the more important one of the two is the systemic circuit. Now, systemic refers to AACVV. The first A is artery, second A is arteriole, C is capillary, the first V is venule, and the second V is vein. So I'll be referring to it in Chapter 21 on blood vessels as AACVV. Next slide. On the next slide, we see basically what I said a moment ago about the AACVV. Now, it turns out that arteries are named solely based upon the fact that they carry blood away from the heart. So whether they are oxygenated or deoxygenated is not the reason. So in high school biology, they often mention arteries are red, veins are blue. That's not true. Arteries were named solely based on the fact that they carry blood away from the heart. Veins, on the other hand, carry blood back to the heart. So we do have arteries that happen to be oxygenated and red, but we also have arteries that are deoxygenated and blue. So again, don't do it the old high school way, but rather understand that arteries are named based upon blood carrying away from the heart, and veins are carrying blood back to the heart. And of course, we have capillaries in between the AACVV. Next slide. We have capillaries. Now, capillaries involve the exchange of gases, nutrients, and wastes. Now, we mentioned in the last lecture series of Chapter 19 on blood that capillaries and red blood cells have about the same diameter, roughly 8 microns. So capillaries are important from that perspective, that blood will move, and when I say blood, I'm talking about red blood cells, will move through the capillaries in a single file manner because the diameter is roughly that of a red blood cell. And they'll move slow. So they're going to slowly exchange gases of oxygen and carbon dioxide based upon diffusion. Okay, next slide. This picture is on page 670. Have a general understanding of this picture. Next, we have this slide. Now, this slide represents the 4 chambers of the heart. It is a good idea to know the 4 chambers. Now, to memorize this particular slide is not the best way. You really do need to apply this slide to a model. On lab day, you'll be doing just that, applying this slide to a model. Next slide. Next slide talks about the heart's basic anatomy. The tip of the heart pointing towards the left arm is called the apex, whereas at the top of the heart, we call it the base. So it's sort of contradictory of what we might expect. So the top of the heart is considered the base, and the pointy tip at the bottom pointing towards the left is the apex. Now the heart is actually surrounded by a pouch, a sac, sort of like a saran wrap. And that pouch is called the pericardial sac. So the heart is surrounded by the pericardial sac, or a pouch. Likewise, the word mediastinum that you see on the slide represents the sort of a cushioned pocket for where the heart would sit in the chest cavity. So yes, the heart fits in the thoracic cavity, but it fits in within the space called mediastinum. Okay, next we're on page 671. And on page 671 you see this picture. Again, we see our major areas. We see the pericardial sac surrounding the heart. It is even labeled for you partially. So we have the pericardial sac surrounding the heart, the tip of the heart being the apex, and the top of the heart being the base. Next slide. The next slide is talking about the pericardial sac. Now, of course, when you have a pouch, you have an outside cover and an inside lining. Well, in this picture, we're going to see a term, or in this slide, we're going to see a term called parietal pericardium. That is actually the outer part of the pouch. So it's actually the outer part of the pouch. Then we have a term called visceral pericardium. Now, the tricky part of this is that even though we call it the inner layer of the pericardium, this visceral pericardium is actually also called epicardium. Okay, so let's make sure we're clear on this. So visceral pericardium is the inner side of the pouch, but it's also called epicardium because if we actually took the pouch off the heart, the outer lining of the heart would be called epicardium. So in the textbook, they make that comparison and say, if we looked at the heart itself, the outer covering of the heart would be epicardium, but the same epicardium is the inside of the pericardium. So that little trickiness is involved here. Okay, one last time. So the outer covering of the heart is epicardium, which is really the same as visceral pericardium. And then the outside layer of the pouch would be called the parietal pericardium. Now this pouch fluid has pericardial fluid. So this pericardial sac has pericardial fluid. It's designed to help the heart cushion itself when it is beating. Now the same sac can often get infected with bacteria and have what's called pericarditis. And when you have pericarditis, it is actually an infection with fluid swelling, and it does not allow the heart to pump well. Next slide. Here's the slide that mentions the pericardial fluid. Notice the purpose is to help stabilize the heart because it cushions the heart as it beats. It is the same fluid that can be infected, and then it would cause a problem with the beating of the heart. Next, we have this picture on page 671. And this picture indicates a general flow of what things are, where things are. Have a rough idea of this picture. Okay. Next. This picture is also on page 671 to show you what I was saying earlier. See how the author has the heart itself shown? The heart itself is monitored within this region. And now see how the author calls it epicardium. And then the outer part of the pouch is called pericardium, or the parietal pericardium. You do not see that visceral pericardium because it's the same thing as the epicardium. So the outer covering of the heart, which is epicardium, is the same as your visceral pericardium. Okay, next. This slide is just to let you know that the chamber of the heart is called atria, whereas the outer, what looks like an elephant ear, and you'll see that on the model, hence the reason for watching the models on lab day, is that the elephant ear looking thing on the side or outside is called the auricle. Okay, now, just as in 2401 you had words like sulcus and gyrus when we had the brain model in 2401, we have little valleys on the heart itself, and those valleys are called sulci or sulcus as singular. Now, you will need to know these terms on the model itself. And the author has done a very good job by saying that blood vessels actually follow the sulcus. So blood vessels actually follow along the groove of the sulcus. Okay, next slide. This is what I was saying that it's really important to know the model. This slide is found on page 673. Page 673. you will need to know the items that are labeled on this slide. Now in the lab days, make sure you actually hold the model of the heart and study these locations. I'm going to go around the clock on this picture. First, at the top, we have the left subclavian artery. This is a branch off of the aorta. The left subclavian artery will go down the left arm. So that's the first item, branching off the aorta. Then we have the arch of the aorta itself. Notice it's broken down into the ascending portion, the arch portion, and then the descending portion. So ascending, the arch, and descending. Next, we have this term called ligamentum arteriosum, and that's this piece right here. Make sure you know the before and after about that term, ligamentum arteriosum. Before you're born, it takes on a different name and a different function, a different purpose. After you're born, it takes on this thing called ligamentum arteriosum. Hence, there used to be a flow here, and now it turned into a ligament type of material. So there was a vessel connecting the two before you were born. Next, we have the left pulmonary artery. Now, this is why I said that you should give away or lose that old high school thinking about arteries are red and veins are blue. That's not the case. Left pulmonary artery, this piece right here, is an artery because it leaves the heart. However, it is deoxygenated, so it's going where? It's going to the lungs. Okay, so that's why arteries are not necessarily named based upon oxygenation, deoxygenation. Rather, they are based upon leaving the heart. Then we have the pulmonary trunk. Next, we have the auricle. As I mentioned, the auricle is an expandable surface. It looks like an elephant ear. It is not the chamber. See how the author says, auricle of left atrium. The chamber atrium is underneath this. Next, we have these yellow fats, and we also have the groove. And the groove is named, in this case, anterior interventricular sulcus. There's a groove, best seen in the model. We have this side being the left ventricle chamber inside, and then we have the right ventricle on this side. Then we have the next groove, which is your next coronary sulcus. Then we have the right atrium with its auricle. So this is the auricle itself, and the right atrium would be inside of it. Then we have this particular vein. And notice I said vein, but yet it says superior vena cava, because that's just a fancy, glorified version of a vein. So superior vena cava. Then at the bottom here, you're going to have the inferior vena cava, not shown. So superior vena cava, inferior vena cava. All right. Okay, now let's go back to the top. We have at the top the brachiocephalic trunk. That's the oddball one because there is not a symmetry here. Here's what I mean by that. That brachiocephalic trunk will actually go and break off into a piece. One piece will become the right subclavian artery going to your right arm, and the other piece that breaks off later will be your right common carotid. So this brachiocephalic trunk breaks off into the right common carotid and the right subclavian later. Then the middle one, our last one, is the left common carotid artery. So the left common carotid artery goes directly to your neck and head. Next slide. This slide represents the actual anatomical picture version. Next, we have the back side. Same story applies, but this time around there are certain things that I want you to note. On the back side, you see the superior vena cava and now the inferior vena cava. That's one thing new that's on the back side. The other things that you notice that are new from the backside view is that you see 1, 2, 3, 4 pulmonary veins. So 2 over there, 2 over there. So you have 2 left, 2 right. Now notice it says pulmonary veins. So these 4 pulmonary veins lead the blood back to the heart from the lungs after they've been oxygenated. So veins in this case are carrying oxygenated blood. These are now the 2 arteries. Last time you could see one, and now I'm showing you both of them. So we have the left pulmonary artery, the right pulmonary artery, both of them leading to the lungs, and they're deoxygenated. Now the heart itself has its own circulation, we'll see, and you see this area right here, again a groove, therefore it's a sulcus, and then you see this thing right here called coronary sinus. That part right there actually bleeds into or feeds into the right atrium. So when the heart itself is done with its blood, the deoxygenated blood from the heart itself will go from the coronary sinus into the right atrium. Okay, next slide. This slide simply lets you know where the heart fits in terms of the rib cage. Have a general idea of where the heart fits in. Notice most of the heart is found on the left side of the body. So most of the heart is on the left side of the body. The other thing you want to notice is that the left side, the part that has the apex of the heart, the left side is going to carry the oxygenated blood, and the right-hand side is going to carry the deoxygenated blood. So there is a sidedness to the body. Next slide. Next slide goes with the heart wall and the different layers. We've already mentioned this one, the epicardium. The epicardium is going to be the outer covering of the heart. Now, it does not involve, that's why we have a different name, it does not involve the pericardium. So the outer covering of the heart is epicardium. True, it is considered also the visceral pericardium, but in this case, we're only talking about the heart itself. Then the next layer is the myocardium. Now the myocardium is the one that we use all of those terms that we learned in 2401, such as the intercalated discs, such as gap junctions. So this myocardial layer is the one that is really responsible for the electrical stimulation through the heart. Next, we have the endocardium. The endocardium will play a role in terms of the lining of the chambers. So the lining of the chambers. Next slide. This is what we said earlier, so I'm going to pass this slide up. It is a good idea to look at it, but we've already covered it. So we said epicardium's other name is visceral pericardium, and it basically covers the heart. Next slide. Next slide we have the myocardium. Now this is the slide that goes with our nice myocardial layer dealing with the gap junctions and the electrical stimulation by way of the intercalated discs. Then at the bottom we have endocardium, which we mentioned is the lining of the chambers. Okay, now next slide. Here's what the picture version of that looks like. So here's our epicardium right there. Here's our sac portion. So this is where the fluid would be. That's my epicardium there. That's my pericardial sac, and the fluid would be sitting right there in the middle. This is my myocardium, and we have our endocardium on this portion. Okay, now, next slide. This slide shows you that the heart is actually in a sort of a circular or oblique or diagonally wrapped around. Now what this does is when the heart actually pumps, it actually has a sort of a turning motion. So it's sort of a twisting motion at the time that it beats. Okay, so there's a Every time the heart beats, there's a sort of a twisting motion. Next slide. This slide talks about the stuff we just mentioned. So yes, the cardiac muscle has intercalated disc from 2401. It's got our gap junctions and is responsible for the action potential going through the heart. Next slide. On this slide, we have our intercalated discs shown. In this case, it's been cut. Next, we have another view of the intercalated discs. And we have our slide version of the same thing, showing us our intercalated discs. And of course, we would have gap junctions in between. And that is what allows the cardiac muscle to act in unison, to act as one. Next slide. Okay. We're going to pass this up, but it is something to keep in mind. So make sure you're aware that cardiac muscle has a single central nucleus. And of course, it has intercalated discs. So that we've learned before. So single central nucleus. Next slide. This is a comparison. You've had this kind of slide in 2401. So whichever book you had in 2401, you had some kind of comparison between the cardiac muscle and the skeletal muscle. Go back and review that. Next slide. Here we have our internal view. Now we have the interatrial septum, which separates the left and right atria. And we have the interventricular septum, which separates the ventricles, the left and right. Now this is the same type of wall that actually can damage during childbirth. So during birth defects, if a child has a birth defect, they have sometimes a hole in the septum that separates the atria, and sometimes they have a hole in the septum that separates the ventricles. So you often see that in birth defects. Then we have this picture. I'm going to pass up this slide because I'd rather explain it from the point of view of the picture. Now I will say that we have several valves. We have the atrioventricular valve, which is the AV valve, and we have the semilunar valve, which will come up on a later slide. So we have 4 valves. 2 of them are characterized as AV valves. The other 2 are characterized as semilunar valves. Now, before I go forward, I'm going to mention something that will be brought up several times later, and that is these valves open and close solely on the basis of a pressure gradient. That's a very important phrase to keep in mind. So when the heart valves open, they open because of pressure gradient. Next slide. The next slide, again, nice to look at these slides. I'd rather show them from a picture point of view. So we're going to pass that up. Next slide. Now, this is what I mentioned about structures. Here's one structure. Okay. When you are an infant, okay, when you're an infant, you have this. When you are a fetus inside mommy's tummy, this is what you have. You have a foramen ovale. That's actually a hole. And you're going to find this foramen ovale in the right atrium. Now, once you are born, it says seals off at birth. Once you're born, that foramen ovale seals off, closes off, and it now turns into the fossa ovalis. Now, we used to have a connection that is now sealed off. So there used to be a connection from the right atrium into another region, and that went from foramen ovale to fossa ovalis. Make sure you see it in the lab day model. So when you're doing the lab day, make sure you look at that. Okay, the pectinate muscles, again, I'll show you that in picture form. And let's go ahead and go to that picture. So here's now the picture I will now describe. Some of the terms have already been mentioned, so they will not be repeated. Others have not. So let's go through this. We're going to start off with this term and then move our way down since others have already been described. So we're in the left atrium. From the left atrium, I pass through this valve. The valve is called mitral valve or bicuspid valve. So from the left atrium, I pass this valve, which is mitral valve or bicuspid valve. I then enter the left atrium. Now, one of the key things about the left atrium is that you see the thickness in the wall. That is a very thick myocardial layer. That's a very thick myocardial layer. Let's compare that briefly to the right-hand side, and you'll see that the myocardial layer on the right is very thin. Now, logic would say, why? Here's the reason. The left ventricle has to pump the blood all the way around the body and have enough force to get you back to the right atrium. On the other hand, the right ventricle, this side, the right ventricle is only sending it to the lungs. So there is a difference between the two. There's also a difference in the amount of force or pressure created. Okay, I'm now in the left ventricle here. Then we already talked about the two little walls. One wall was the interatrial septum, and the other thicker wall is the interventricular septum. Then we have this trabecule, which sort of helps turn the blood and turbulate the blood inside the lining of the chambers. That's the purpose of that tubercle. Then we have on this side, we have our inferior vena cava I've mentioned, and we have our right ventricle. Now the right ventricle is going to push the blood out through this valve called the pulmonary valve, and some people call it pulmonary semilunar valve. So the pulmonary valve or semilunar valve, and that's going to get you out into the pulmonary trunk. Now let's go over the heart valves again. So I'm going to start up here now. The first thing we see here is the fossa ovalis, which I mentioned in the previous slide was formerly called foramen ovale, right? So the foramen ovale became fossa ovalis. Then we have the opening of the coronary sinus. I mentioned that in a previous slide, that the coronary sinus leads into the right atrium. So here we are in the right atrium. The right atrium has a valve, and you pass through this valve, and that's your tricuspid valve. And the tricuspid valve will get you into the right ventricle. So far I've described this valve, and that one. There's one more missing, and that's right here in the corner, called the aortic valve. The aortic valve is an important one. The aortic valve has little rings on the valve itself, and the two rings are actually openings. And those two openings lead into the pulmonary area. They actually lead into what's called the coronary blood circulation. So they're near the pulmonary area, but they're going to lead into the coronary blood circulation. So you'll actually see the coronary blood supply coming right off of the aortic valve. So the aortic valve has two openings, one for the left coronary, one for the right coronary. Your book does not provide that picture, so I'm giving it to you this way. So again, the aortic valve has two openings, one for the left coronary and one for the right coronary. I'll mention it again in the correct place. So that's going to be part of that aortic valve. Then once the blood is past the aortic valve, it ends up into the aorta. Now the heart's job is really just to get the blood out past this aortic valve. And while the heart is actually resting in between beats, the blood is trying to regurgitate back into and past the aortic valve. And that is where your AACVV, your blood vessels, come into play. Your blood vessels have a job of getting the blood away from the heart away from that aortic valve. And that's what they do in between the heartbeats. Okay, so that's the main gist of this picture. The two other things I need to cover are the chordae tendineae and the papillary muscles. The purpose of the papillary muscles is to hold the chordae tendineae. Now why? Because you do not want the heart valves to invert like a cheap umbrella in a stormy wind. So as an umbrella will invert the other way, you do not want that to happen to the heart valves. The heart valves should always face in a given direction and then close. So you don't want that umbrella flipping the other way. You don't want the heart valve to flip the other way. So we have chordae tendineae that does that along with the papillary muscles. Okay, that takes care of this slide. Okay, much of what I've mentioned is already here. So the chordae tendineae, papillary muscles, as I've said, prevent the valve from opening backwards, like a very strong wind with an umbrella. Okay. And again, all these valves prevent what? Backflow. So we're trying to move the blood in one direction. Next slide. I've already mentioned this part right here. The trabecule, no major thing. It does play a role with the movement of the heart itself. So it's more of a structural role. Next slide. Next slide shows you the papillary muscles and the chordae tendineae a little bit better, preventing and ensuring that the heart valve stays in one direction. Okay, next slide. These are the pulmonary circuits. We've already talked about them. We're going to go to the next slide. Again, we've already talked about these. I mentioned that the left atrium to left ventricle, to get there, we have to pass the mitral valve. The other name for that is bicuspid valve. Okay, left ventricle, I've already mentioned much of this too. It holds the same volume as the right ventricle. It doesn't seem like it does, but it does. It has a thicker wall. We've already talked about that. It is more powerful because it's got to push the blood all the way out of the heart, around the body, and back to that right atrium side. Okay, so we did mention that too. Next slide. The aortic semilunar valve, or the ascending areas, this slide we've already mentioned as well. Don't care for that picture. Okay, now we go to this one. Now remember the rule, and we've already discussed a lot of this too. The right ventricle has a thinner wall, and it has less pressure than the left ventricle, and we've already talked about that as well. Don't care for that slide. Don't care for that slide. And now we're on this slide. Now, I've already mentioned to you that the heart valves are one-way valves. They prevent backflow. And remember, their job is to do what? Move the blood further along. And what opens and closes the valves? The pressure gradient. The differences in the blood pressure will do that. Next slide. We've already talked about that. This slide, we've already discussed this as well. This slide, now this is the one that I mentioned, that the aortic valve has tiny little holes, and I did say that this book does not show them. So the aortic valve has tiny little holes, and they're allowing the coronary blood supply to come out. Not shown. Now, starting with this slide, we're going to notice that the author is trying to show us how the blood moves and which valves are open and which are not. Now, one of the things you're going to have to understand is that whenever we say that a chamber is relaxing, whenever a chamber is relaxing, it equals filling. Whenever a chamber is contracting, it equals emptying. So that's an important phrase to remember from this point forward. Now on this slide we see the left atrium receiving the blood. We also see that the left ventricle must have done something because of the side banner. What do you see on the side banner here? It says that the relaxed ventricles. If something is relaxing, the chances are something is contracting. There are only two exceptions to that rule, but typically, if something is contracting, something must be relaxing. So in this picture, we see that the relaxed part would be the ventricles. So the ventricles are relaxing, implying that the atria must be contracting. So when the atria contract, we get the blood to move in. And as the atria contract, what happens to our AV valve on the left, which is called the bicuspid valve? It opens. Remember, a pressure gradient. So when I squeeze, I have higher pressure here, less pressure here. That causes the gate, or this, in this case, the valve, to open. That allows the chordae tendineae to loosen up. The papillary muscles also relax and allows the blood to move in. Here it says left ventricle relaxed and filling. Remember, relaxed always goes with filling. And while this is filling, what is happening here? The aortic valve is closed. Now, of course, when it's closed, what must be happening to the blood up here on top of the aortic valve? It must be regurgitating, shown by the arrows here. Right? And of course, whose job is it to make sure that this does not regurgitate back? The blood vessels, the A, C, V that I keep talking about. So the blood vessels will be moving the blood forward at this time. So that's picture A. That's page 679. Okay, continuing. This is just another view of it. And we go to the next slide. Okay, just another view of it. Here's the other part now. Notice the side banner. This time, who is contracting? The ventricles are contracting. So here's what's going to happen. When the ventricles contract, they're going to, of course, empty. Now, when I push up, what's going to happen? The papillary muscles will contract, the chordae tendineae will tense, and of course, the bicuspid or the mitral valve will close. Remember, the valves are one-way direction only. So when I squeeze, these will close. While these close, the neighboring one, called the aortic valve, will open. Why do they open? Because we have a higher pressure here in the ventricle in comparison to the aorta. So high pressure, low pressure, from high to low. So the contraction or squeezing of the ventricle is going to push the blood out through the aortic valve and into the aorta. Okay? So that's an important notion. Again, it's the pressure gradient. Now, of course, what should be happening according to Nemesh's rule? If this is contracting, then of course, what should be happening in the atrium then? Ventricles are contracting, the atriums must be filling. And if they're filling, they are relaxed. Okay, that's typically how it works. Okay, this slide, pass by. You can read it at your leisure. And that takes me to this. I'm not really too keen on this either, so we can pass that one up. And we go to this. Now, this is where the importance comes in. The blood of the heart has its own circulation. So the heart itself has its own circulation called the coronary circulation. It's made up of coronary arteries and veins. So coronary arteries and veins. So the blood must also go to the heart, and the heart is rich with its own blood supply. Now we have our coronary arteries on this slide. I've already mentioned that the coronary arteries actually start where? Right at the rim of the aortic valve. Here the author calls it originates at the aortic sinus, and the aortic sinus is actually just at the rim of the aortic valve. I keep mentioning it because the author does not have a clear picture for it. What opens that area? Of course, is the pressure. Okay. Any questions on those? Then we have the right coronary artery. So we have the left and the right. We're starting with the right. Now this slide I would make a note of, but you really need to be able to see it from the heart model itself. So we're going to pass this up, but make sure you look at this slide when it comes to looking at the heart model. Same is true for this slide. It's only going to make sense if you study it on the heart model. Okay, for this one, it is important to know where these two are located. The circumflex artery and the anterior intraventricular artery. Make sure you know where they are. So circumflex artery, anterior interventricular artery. Make sure you know where they are. For the bottom half of this slide, I really don't care too much about it. Just I want you to know that this term right here, anastomosis, simply means branching. So anastomosis simply means branching. Next slide. Next slide, we have the cardiac veins, which are basically the return back to the heart. Same is true for this slide. Make sure you know the terms, but make sure you know how they apply on the heart model itself. Next slide. This is what I'm talking about. Can you apply these terms to the actual physical model? Now, one of the rules of making this work for you is taking the model of the heart on lab day and comparing it to this picture, and how many can you get right. Now, when it comes to the blood vessels, they follow what? The sulcus. So they follow that sulcus, they follow this sulcus, and they follow that sulcus. So again, make sure you know the coronary circulation, both for the arteries and the veins for the heart. Here is the next slide for the back side of the picture. Again, having this in front of you while you're studying the heart model would really help. Yet another. Now when we go to page 682 and 683, there is a little spotlight. And this book calls it spotlight or focus or whatever. And it talks about the coronary artery disease. Have a feel for page 682, 683. No heavy detail, but just make sure you've read that section. Okay. So the slides I'm now going through are mentioned in that reading on page 682, 683. Okay. So angina pectoris, myocardial infarction, these are mentioned on those sections, 682, 683. Okay, that takes care of that. So again, these are all mentioned there. Here is what happens when you have a heart attack. This is a blood clot, is what they call it, occluded, and the areas below which would have been served are now dead. Okay, draw a line. Now that starts off the next main topic, and the next main topic is your heartbeat. And that is found on page 684-ish. Now, the heartbeat, we have the atria involved and the ventricle involved. So first the atrias, then the ventricles. And that makes sense. Next slide. We have the cardiac physiology in terms of conduction system. Now, I really don't care too much about this slide. This is the terminology the book is using, but be aware that we have a conducting system and a contractile system. Now, this slide is important. We have our SA node. The SA node is the initiation of the conduction system. So your heart cycle starts with the stimulation of the SA node. Now, when you have the SA node triggered, you're basically telling the heart electrically and chemically to do what? Stimulate. So the SA node is triggered either chemically or electrically. That is a slide that's not here, but you should be aware of. So the SA node is triggered electrically or chemically. Now we have the EKG or ECG at the bottom of this slide. This is a recording of how the heartbeat went through electrically. Now the EKG is not an absolute measure. It can be fooled easily. So when you do take an EKG, you have to take it multiple times just to see whether the episode is still there. Often some people have issues with the EKG only at the time of that incident. And then once that incident is gone, the EKG returns back to normal and no one is the wiser. So it's important to understand that the EKG only will record what's going on at that moment. So if you have a sporadic issue, you may not catch it. Okay, so it's not a tell-all for everything. Then we have the conduction system that we talked about a moment ago. Remember the heart, it's automatic. It has rhythmicity, right? So the heart is automatic. It has rhythmicity. Okay? That means the heart will beat on its own if it's given the right nourishment and nutrients and environment. It can beat on its own outside of the body. Okay? So it doesn't need the nervous system to actually stimulate it. It has its own rhythm built in. Now we talked about the SA node, and now we're talking about the AV node. So where do I find the SA node? In the wall of the right atrium. Where do I find the AV node? Just between the areas of the atria and ventricle, where they both meet. So the junction between the atria and the ventricle. Don't care for that slide. I really don't care too much about this slide except to let you know that what comes first, the SA node. And it's the SA node that actually acts like a rhythm center and a rhythm setter for the heart. So the rhythm center and the rhythm setter. So when you're playing, say, a relay race, who really sets the rhythm? The very first person that runs with the baton is setting the rhythm, the rate for the rest of the team. It's just like that. So the SA node will depolarize first and set up the heart rate. Next slide. Here's our SA node, again in the right atrium. Here's our AV node right about there at the junction of the atria and the ventricle. We then have our AV bundle and then our bundle branches. Some people call it bundle of His, H-I-S, named after someone. So bundle branches. And then we have our Purkinje fibers headed back upwards. Okay. Now why is this slide so important? This slide is important because the physical story is related to the electrical story. So just as I keep talking about electrical and chemical all the time, the story that goes with this slide is that electrical first, then physical. So how does that work? It works this way. If I'm stimulating the SA node electrically first, then that SA node is going to translate to the contraction of the atria. So again, if I trigger the SA node, then I'm going to trigger the atrias to contract. If I get down to the AV node and then move down the bundle and I get all the way down to the Purkinje fibers, then what's going to happen? I'm going to stimulate the ventricles electrically first, and what will follow? The contraction, the physical contraction of the ventricles. So you see how that works. Electrical before physical. Now that line, that little catchphrase of electrical before physical, is actually what shows up on an EKG. Okay, so that's exactly what shows up on the EKG. Now, I don't want you to memorize this slide that we're now looking at. I just want you to understand that who is setting the heart rate. We said the SA node. The SA node is being set by the sympathetic nervous system and is being monitored by the parasympathetic. Now, what does that mean to us? It means that if the parasympathetic had no influence on the heart, then the heart would actually beat at a much higher rate. So the SA node is actually being curbed by the parasympathetic. So if I let only sympathetic kick in, the SA node would beat at something like 100 to 200 beats per minute, way up there. But it is the parasympathetic that's actually curbing the action of SA node and the sympathetic and keeping it at around 74, 75 beats per minute. That's an important statement. Okay, next slide. Okay. Now, having said what I've said, the slide now says it too. It says the SA node would generate at about 80 to 100 action potentials per minute. The AV node would be around 40 to 60 action potentials. And whose job is it to actually slow the heart rate down? It's the parasympathetic. So the parasympathetic is actually curbing the action of sympathetic on the SA node. It is slowing the heart down for us. Okay, this slide we can pass up. We already know where the pacemaker cells are. The pacemaker cells are your SA node. So the pacemaker setup is the SA node. Next slide. Here we go again. We now see the stimulation at the SA node at time 0. Then, what's going to happen next? In the next slide, and I'm going to show pictures only, in the next slide, we have this. Notice we have not got to SA node yet, or rather the AV node yet, right? So we haven't gotten to the AV node. So the SA node is stimulated, but not the AV node yet. That means that the top portion has not yet contracted. Not yet. So the top is about to contract because it hasn't got to the AV node yet. Next picture. This picture shows you that we've pretty much done what? Got the entire atria section colored here, which means that we're now going to start stimulating the AV part. We haven't done that yet, but we're starting to stimulate the AV part, the AV bundle. Now, what is happening in this physically? Remember how I said electrical before physical. While this is happening, we're about to do what? Contract the atria. And when the atria actually are contracting, what's still going down this septum? Let me say that again. What is happening while we're still contracting at the top? The electrical signal is going down the bundle, down the septum, while we are emptying into the ventricle. So yes, as we fill the ventricles, the electrical signal is actually going down this septum wall. This bundle branches, the AV bundle. So yes, the electrical signal is reaching here before we're ready to contract. Okay, next slide. This is number 4 now. In number 4, what do we see? We see the bottom still not contracting because it is actually doing what? Filling. So the bottom is still filling while it's being electrically stimulated. Once it gets to the Purkinje fibers, the bottom will be ready to contract. And now we're here. Now watch this. I'm going to read this one carefully. The impulse is distributed by the Purkinje fibers and relayed throughout the ventricular myocardium. As you can see, it says atrial contraction is now complete because we did what? We squeezed the atria, we filled the bottom, and while we were filling the bottom, let's say that importantly again, while we were filling the bottom, the electrical signal was getting there. And then once we fill the bottom and we've got the electrical signal there, now the bottom is going to contract. And notice how it says it here, ventricular contraction begins. That's the magic of the electrical and the physical story. So again, the top squeezes. And while the top is squeezing, the bottom is filling with blood. While that bottom is filling with blood, that time delay allows us to also get the electrical signal down to the ventricle. That's why there's a delay between the SA node and the AV node, giving the ventricles time to fill. And once the ventricles have basically filled, we have already done what? Got the electrical signal down there, and it's now ready to contract. Okay, next slide. Bradycardia, tachycardia. So bradycardia, tachycardia. Bradycardia is slow heart rate. Tachycardia is fast heart rate. Now, these four at the bottom are reasons why your EKG would get messed up. So if you have a damage, a disease, an injury, whatever it is, it's going to relate to these four. One, abnormal cells. Some of the cells may be died. They're dead. We have some problems with those cells. You might not be able to pass the electrical signal along. So the neural system within the heart may be dead. The bypass conducting system, so the AV bundle or the bundle of His or the Purkinje fibers may not work. Or we have a disruption in the ventricle contraction because the heart muscles are dead. So there are these four reasons why you have a problem with the conduction. Next slide. We've already talked about this, but let's go over it real quick. We said the abnormal patterns diagnose damage. So if you have an electrical damage in the heart, and as we've said, electrical follows physical, right? Electrical then comes physical. If the electrical is damaged, you're going to have a physical damage to the heart as well, because electrical represents some physical damage in the heart, and then therefore the heart's not going to function. So electrical before the physical. Now let's look at a typical EKG. Here are the waves that you find on an EKG. This slide is important. So we have a P wave, a QRS complex, a T wave. Now words like depolarize, depolarize, repolarize, they're all referring to the electrical part on an EKG. But of course, once you understand the electrical, what will follow? The physical. Same comment applies to this slide for intervals. So the interval story is important as well. Now on the bottom of page 688 is where this section is found. On the bottom of page 688. Make sure you read that entire left-hand column of page 688. It talks about what happens to these intervals. So page 688, the left-hand column. Now we're going to briefly go through what an EKG looks like. So here we have one heartbeat and then another heartbeat. So two heartbeats are fully shown here. One heartbeat, two heartbeats. And here's the enlarged view of it. Now, first we have a flat line to talk about. Anytime you have a flat line on an EKG, it means something is not contracting. So a flat line represents Something is not contracting. Basically what? Something is relaxing. Okay? So if it's not contracting, it's relaxing. If we had only a flat line throughout this entire picture, then you know you'd be dead. But what does it represent? It means that you have no electrical conduction going through the heart, which basically means that the heart is permanently relaxing. Okay? And there's no stimulation, no depolarization, no stimulation of the ventricles or atria to contract. Now let's go through the picture. So here we have a flat line and then a bump. This bump represents P wave or atrial depolarization. While I have this bump, it means that I am sending electrical conduction through the atria. And while that's going on, so I have this bump, we see a flat line here. It means that while I have this going on, the bottom of the heart is now doing what? Resting. So you could translate this picture as what? Depolarization is related to atrial contraction. Okay, so atrial depolarization related to atrial contraction. If that is the atria contraction, then this must be ventricle relaxation, this flat line. Then I have the QRS. That means that the ventricles are being stimulated. And while the ventricles are stimulated, I have this flat line. In this flat line, it's the ventricles that are contracting, but the atrias that are relaxing. And then I have the T wave. The T wave is when the ventricles are repolarizing. So the ventricles are resetting. Now, hidden within this, and it is mentioned in the reading of page 688 and the page before, it is mentioned there where you don't see an atria repolarization because it is actually covered during this stage. So it's masked. It's covered during this stage. So it's masked. So the T wave represents ventricle repolarization. The ventricles are relaxing and the atria are getting ready to reset. While that resetting is going on, we have a pause. That pause is when all four chambers are relaxing and the valves are closed. It is basically a rest period between heartbeats. Had I had this picture very close to this one, say right here, instead of having a long space, then I would have one heartbeat followed by another in a rapid succession, and that would be tachycardia. Okay? So be aware of this picture. Now we're going to go through these abnormal ones, the cardiac arrhythmias. I'm on page 689 for this. In this one, we see a P wave, a T, a P, and then we sort of see a T followed by a rapid P. So we have a premature atrial contraction. That's why we have an extra P too soon. Okay, so an extra P too soon. And that's your atrial contraction premature. Then we have tachycardia. Notice I'm not giving any pause like I did in the one above. Here you see a pause between the next P. We don't see a pause, we don't see a pause, no pause, no pause. So in this case, we have atrial tachycardia. It's the top half of the heart that's just pumping away prematurely. The bottom shows you we have a problem with the top being stimulated. So we have atrial fibrillation, where the top of the part of the heart is just fluttering. It's not really contracting well. We do see that the ventricles are okay, but it's the atria that are not doing so well. And in this case, there are cases where atrial fibrillation you can survive from. Next slide. The bottom half of page 689 shows you premature ventricular contraction. Everything's fine there, but all of a sudden we lost a P and we just had a QRS. Okay? We had a premature bottom half of the heart contracting before its time. Here, ventricular tachycardia, all we see is the bottom pumping, 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 pumping faster than the top. And then on the bottom we have ventricular fibrillation, where the bottom half of the heart is pretty much not pumping. It's fluttering, but it's not pumping. Ventricular fibrillation you're not likely to survive from because the blood's really not moving around correctly. Okay, any questions on that? Okay. Now, before I go to the next slide, it's important to note that all of this is made possible because of ion channels. So again, we're going back to 2401 in essence. It's the electrical ion movement that is determining this depolarization, repolarization, just like we learned in the skeletal muscles of 2401. Okay, now, if you look at this slide and you see words like resting potential, you know that we're talking about the ion channels, such as the sodium channels, potassium channels, and whatnot. That's exactly what we have here. Next slide. On this slide, we see, as we did in 2401, we see something that is shooting up. That is your sodium entry, that sodium depolarization that we've learned before. Then we have a plateau. Now this is new. For the cardiac muscle, this plateau is due to the calcium entry, so the calcium ion channels. And these ion channels for calcium are the ones that help determine the contractility of the heart, how strong that muscle, that myocardial layer, is going to contract. So how strong that myocardial layer is going to contract. And it's the plateau due to the calcium ions. Then we have our repolarization due to the loss of potassium. So it's really up to the potassium output. So when potassium leaves the cell, that outflux, that plays a role in returning back to our resting membrane potential. So if you really want to alter the function of the heart, either for good or bad, the object of the game is to mess with the calcium channel and the potassium channel. Okay, next page. The page I'm on now is page 690, and it's a comparison between the skeletal muscle and the cardiac muscle. This slide is worthy to look at. So we do have this nice strong contraction that's maintained by the cardiac muscle, but not by the skeletal muscle. Okay, next slide. I really don't care too much about this slide. Now on this one, I do want you to know that it says 30 times longer. That's where the contraction story comes into play. So muscles of the heart can maintain that. Now remember, we do not have what? No tetany. So cardiac muscle does not exhibit tetanus. So cardiac muscle does not exhibit tetanus. Right, so you will not see tetanus in cardiac muscle. Next slide. Don't care too much about this slide. Okay, this slide is important. Just make a note of it, sort of a summary of it. Next slide. Same is true here. Make a note of this slide. Now, the last two slides I put up that said make sure you look at, they're simply explaining that it's the calcium ion channels and the calcium that plays a role in the contractility, that long stretch, that long contraction that cardiac muscles exhibit, instead of having that fast twitch kind of thing. Now, of course, this slide says that cardiac muscles, aerobic energy of the heart. There's a lot of oxygen needed by the heart. If you really looked at the overall body, I would say that the brain and the heart are very key in terms of oxygen demand. So the heart and the brain are key in oxygen demand. And so you really have to have a lot of oxygen to the brain and that of the heart. Next slide. Next slide starts off the cardiac cycle. So the next slide is cardiac cycle. To best understand it, we first need to understand these two terms on the slide you now see. Systolic goes with any phase that is under contraction. So systolic goes with contraction. Diastolic goes with relaxation. So those words I gave you earlier, relaxation equals filling, contraction equals emptying. We now have technical terms like systole and diastole to go with that. Next picture is this one. The best way to understand this picture is to relate this one to your EKG. We're going to briefly go over this one. First, we have at time 0, we have the SA node being stimulated. So in this top picture, it says atrial systole. So you already know that in this top portion right here, what is happening? We have stimulated the SA node. We're stimulating physically the atria. The valves have opened. So the bicuspid valve on the left and the tricuspid valve on the right have opened, and we're now filling the bottom. So that's the atrial systole. Again, related to your EKG. So this is just before the QRS. So this part right here is before the QRS. Then comes to this point. This point, what are we having? Ventricular systole. So here we're now starting to do what? Contract the bottom of the heart. So EKG-wise, this would be starting off the QRS. So between B and D, right here at C, we should have the QRS on the EKG. During the EKG, during the QRS, what are we doing? We're contracting the bottom. Then there's a moment in time that we're going to have all 4 valves closed. So as you can see, all 4 valves are actually closed. This is actually a moment in time where the heart actually gets to rest. So we are relaxing the ventricle. We're also at the same time relaxing the atria. So this is a brief moment where all 4 are actually resting. All 4 chambers are resting, all 4 valves are closed. And this is the point which we call the isovolemic relaxation. Okay. Let me back up for a second here. This would be isovolemic contraction because I just see a little indentation, and then right about here between this one and this one, not shown, but between this one and this one, I would say it's the isovolemic relaxation. It's not shown there, but that's where it would be. So right about here would be the isovolemic relaxation. And then just right about here would be the isovolemic contraction. And again, they don't show it. So you would have isovolemic contraction here, isovolemic relaxation right about there, and then you would have this picture. In this picture, we're about to do what? Reset. Now, what would this be? On an EKG, that would be that flat line. You see, none of the chambers are contracting. Now, what does that mean, Nemish? It means that we see what? We see some blood leaking into the ventricles even before the ventricles are even being stimulated. We also see blood leaking into the ventricles before the atria actually contract. So yes, there is blood going into the ventricles before the atria actually contract. Okay? So there is blood moving into the ventricles before the atria had a chance to contract. Okay, moving to the next slide. Okay, we've already talked about this. I'll read each one quickly. So what's happening in this first phase? Basically, I said the same thing. Atrial contraction forces a small amount of blood, of additional blood, into the relaxed ventricle. So there is some blood going in. Notice it says additional blood. Then we have atriosystole ends, atrial diastole begins. That means that the top are relaxing while the bottom are contracting. So the bottom's about to start to contract. Next slide. We see that the top is now done and the bottom is now contracting. Okay, so now the bottom is contracting. And remember, what opens up the valves? The pressure gradient. Next, bottom's squeezing more of the blood's coming out, shown here. Next slide. Now, this is where I said that there is a moment in time between the last one and this one. There's a moment where all 4 valves are closed. Okay, so all 4 valves are closed. Next slide. Here we have all chambers are relaxed, ventricles are filling passively. So as I said earlier, there is blood going where? Into the ventricles, but it's leaking in by gravity. So the chambers are slightly open and blood's going in. And this is before the atria have actually contracted. So again, the rule of thumb is to understand this cardiac cycle with respect to the EKG. Okay, now let's move forward. We've already talked about this story here. Rises during systole and falls during diastole. So the pressure will actually change. I've said multiple times what allows the valves to open is the pressure gradient. Next slide. We've already said that the heart rate is around 75 beats per minute. The heart rate will increase. So when the heart rate increases, all of this speeds up. So all phases will shorten, particularly diastole. So when you're running in a race and your heart rate's going up, the time period that the heart gets to rest drops. And that's what this line is saying. The time period that the heart gets to rest stops, especially the diastole period shortens. Now that does translate to, even though your heart goes faster, you may be doing what? Moving less blood out if you cannot establish a pressure gradient. Next slide. We've already talked about these. Now this slide is very important. So this slide is very important. Atrial systole. We already know the systole means what? Atria are contracting. What valves are open? The AV valves are open. What's happening? We're filling the ventricles. Okay, then what happens? When the atria actually stop contracting, the AV valves will close. Now, what have we done thus far? And that's why this slide's important. What we've done thus far is that we have filled the bottom ventricle. Once we stop at this very point, like a freeze frame, if we stop here, we have done what? The top has already squeezed. We fill the bottom. The blood that is now in the ventricle, that blood is now called end diastolic volume. So end diastolic volume. The end diastolic volume is usually around 130 mils. So at rest, the end diastolic volume is about 130 mils. That's important to keep in mind. Next slide. Now, how do I have ventricle systole? Well, the bottom has to do what? Squeeze. Now, there is a moment in time. As I said, there's a moment in time where all 4 valves are closed and we're simply doing what? Sitting there. So all 4 valves are closed, blood's in all of them, and it's just sitting there for a moment. That's isovolemic contraction. It's a very short pause. It's designed to build pressure in the ventricles. Then our job is to eject blood from the ventricles. Now I'll be using the example of the left ventricle. But when I squeeze the left ventricle, blood is going to go out. In the case of the left side, it's going to go out the aortic valve. In the case of the right ventricle, it's going to go out the pulmonary valve. Now, the amount of blood that is leaving is stroke volume. So out of the 130 mils of end diastolic volume, we're going to eject a certain amount of blood. Typically, it is about 60% of the end diastolic volume. Now, wait a second. Are you saying that out of 130 mils of your end diastolic volume, we're only ejecting 60% of it? That's right. So the other 40%, that other 40% is now called end systolic volume. Okay? About 50 mils if we take 40%. So 40% of 130 mils, which is about 50 mils, will actually be remaining in the ventricle. Now, is that a good or bad thing? To some degree, it is actually a good thing. We'll see that a little while later. Now, why is it good to have blood still in there? Because if you can have a small amount of stroke volume and yet still get your work done, then your heart's not working so hard. Okay. So you really want to be able to have the right amount of stroke volume here and there. Okay. So stroke volume does play an important role. Continuing. We have, as the ventricular pressure falls, the semilunar valves will close, and whatever's left in there, as I said before, is the end systolic volume. Now let's summarize all this by way of this chart. On page 694, you're going to find this picture. So page 694 shows this picture. The bullets that you see are of value to you. Now, if you notice carefully, these bullets of 1 through 8 follow what? The EKG. So that's why the EKG will tell a doctor quite a bit of information. Next slide. We've already talked about all the other steps, and now we're going to get to finishing up the last 2. Ventricular diastole. In ventricular diastole, when the bottom half of the heart is resting, notice all heart valves are closed and the ventricles are relaxed. So when someone says, when does my heart get to relax in its lifetime? It does it in between the beats during the diastole period. Next, atrial pressure is higher than ventricular pressure. So what happens? The AV valves open, and as I said before, what will happen first? Passive filling of the atria, and then a passive filling of the ventricles. So 1 through 8 that we looked at, those steps, are the cardiac cycle. Be certain that you study the cardiac cycle. That is the most commonly missed issue in 2402. Next slide. Heart sounds. Make sure you're comfortable with the heart sounds. The first sound that you hear, the lub, which is the louder sound, is produced by the AV valves closing. The dub, which is the next sound, is produced by the closure of the semilunar valves. That information is found on page 695. Okay, now continuing. I don't care too much of the top one, but I do care for the bottom. So anytime you hear someone's heart valves and they hear a sort of a gur 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 sound, which means that there's a regurgitation going on, which means that the heart valves are not closing properly. So you shouldn't be hearing any regurgitation through the heart valves. It's almost like a washer or a gasket not working so well and you have a leak in it. It's a little like that. Okay, draw a line. This slide right here, don't care for. This slide right here, I don't care for, unless you look at the slides before. So if you don't look at the slides before, you can use this slide as a summary. Okay. Draw a line. Technically, I'm now on page 695. The formula that you now see on the screen, you must know. You'll need it for the test. So make sure you can define what EDV is, ESV, stroke volume, the formula is there, and the percentage of ejection there. Now, stroke volume is related to what's coming up next, called cardiac output. So that's coming up soon. It's really important that you understand that cardiac output is related to stroke volume and your heart rate. If one doesn't work, the other one's going to have to make up for it. And we'll see that in the formula coming up. Now here's the formula that I'm mentioning. Cardiac output. The volume pumped by the left ventricle in 1 minute. Now what's cardiac output? It's measured in mils per minute. Heart rate is measured in beats per minute. And stroke volume is measured in stroke volume, which is mils per beat. So if I were to say that my heart rate drops, if my heart rate goes down, my stroke volume better do what? Go up to give me the same cardiac output. If heart rate goes up, stroke volume goes up, then I'm going to get a very high cardiac output. And that's usually in the case of exercise. When you're heavily exercising, the heart rate's going to go up and the stroke volume will go up. Now, if you're an athlete, the heart rate may go up, but the stroke volume actually may go down, giving you what? The same cardiac output. Now, there's a rule to this. The rule is what goes out of the heart must eventually come back to the heart. So it's really important that what goes in the heart comes back to the heart. Okay. That means you do not want any pooling in the body. So if you have too much blood leaving the heart and not coming back to the heart, then you might have some pooling in your feet, in your lungs. Of course, we call that pooling edema, which is not a good thing. So cardiac output is going to help us understand this pooling business. Now, on the next slide, you're going to come across this one. It says, cardiac output adjusted by changes in the heart rate or stroke volume. Now, in this slide right here, it says heart rate adjusted by the autonomic nervous system or hormones. What does that relate to according to Nemish? Nervous system being the electrical, hormones being the chemical. So how do I adjust heart rate? As I said earlier, I adjust the heart rate either electrically or chemically by adjusting the SA node. So if I adjust the SA node according to chemicals, like hormones, or if I zap the heart with electrical wiring or whatnot, it's going to do what? Adjust the heart rate. So electrical and chemical affecting the SA node for the heart rate. And then I can affect stroke volume by either letting more fluid stay in the heart or having less fluid stay in the heart. Right, so that's how I can adjust the stroke volume. The formula and this picture go hand in hand. So this slide right here is related to the last one I just showed you. Okay. Now we look at the nervous system. Yes, the nervous system plays a role in the heart. This slide, make sure you make a note of. Again, vagus nerve, cranial nerve number 10, carries the parasympathetic. So the vagus nerve, cranial nerve number 10, carrying the parasympathetic. And as I said, it is the parasympathetic, noted down here, the parasympathetic that's going to slow the heart rate down. And where do I find these centers? In the medulla oblongata. You should know this from 2401. Because in 2401, you learned that the medulla oblongata played a role in both vasomotor of your blood vessels as well as the heart rate. So that's a reminder from 2401. Okay. I really don't care too much about this slide other than something we learned in 2401 as well. One of the things we learned in 2401 is that we have baroreceptors and chemoreceptors. Now typically these baroreceptors, chemoreceptors are going to be found in two main places. The carotid, right in the neck, in the carotid body area, we have baroreceptors, chemoreceptors, and also in the arch of the aorta. So moving to that picture, we see this. This is one area right here in the arch of the aorta, which we'll see in the blood vessel chapter. We're going to see baroreceptors, chemoreceptors there. We also see them in the neck, and they don't show that part here, but they're also found in the neck. So the baroreceptors, chemoreceptors, and the carotid sinus in the neck. So this is your medulla oblongata having the acceleration and the inhibitory through vagus nerve. Okay? So be aware of this picture. Okay. This slide, don't care for. This slide, I don't care for. Now, this slide we've already talked about. I said that I can affect the SA node, either electrically or chemically. We've also said that it's going to be the parasympathetic that slows the heart down. So we've already talked about that. Now, how do I slow the heart down? In 2401, you came across this term called hyperpolarization. Well, that's how I slow it down. I ensure that the heart does what? Breaks, slows down. So hyperpolarization is very much like applying the brake to the heart rate. Sort of like those rides at many of the amusement parks where they have cars that don't have brake pedals. All they have is a gas pedal. And you either apply the gas to go forward or you slow it down by releasing the gas pedal and the car stops. Well, this is exactly like that. So hyperpolarization is like taking your foot off of that type of car where removing your foot off the accelerator automatically stops the car or slows the car down. That's what this is like. Okay, draw a line. Make sure you're aware of this slide. So make a note of this slide. Now on this slide, the only thing I care for is venous return. Now why is venous return so important? Because the amount of blood coming back is going to be related to the amount of blood leaving the heart. Remember the rule, what goes in must come out. Okay, now comes hormonal effect. Again, as I mentioned, the heart rate by way of SA node chemically. And these hormones play a role chemically on the SA node. They either speed up the heart, and typically they do, because in this example we're only talking about what? Sympathetic. So all of these will speed up the heart. Someone that has a high thyroid action, for example, will have a higher heart rate, higher metabolism. Okay, draw a line, new topic. We have already talked about this as well. The EDV is the blood that's left over in the ventricle. It is going to be affecting the filling time and the venous return. So it affects the filling time and the venous return. So if you get more blood out, more blood's going to come back. You get less blood out of the heart, it's going to affect the filling time and coming back. Okay, now, the next few slides I'm going to put up are related to another slide. So let me go there first. This is the slide that we're looking at. And the ones I passed up, I'm coming back to once you make a note of this one. There are 3 factors that affect stroke volume. And that's what this is. 3 factors affecting stroke volume. Now the author says ESV. Well, if you remember the formula, stroke volume is related to ESV and EDV. So these are 3 factors affecting stroke volume. The first one is preload. Let me give you an example of that. Whatever blood is left in the ventricle, we said it was called EDV. And it was the 40% or so that was left there, right? It was related to that because we had EDV first, and then we subtracted by taking the stroke volume out, and then we have ESV, which is what's left over. If we get more out, then we'll have less ESV, correct? Less ESV. So if more comes out, less stays in the heart overall. The heart actually stretches when it has some fluid in it. So if we had some blood left over in the heart and that fluid stretched the heart a little bit, then when it came time to squeezing, the heart, being like a rubber band, will have what? Not only will it squeeze and have a force, but because of the elastic recoil, the heart will have a greater squeeze. So if you had a liquid in a container that actually stretched like a rubber band a little bit, having that blood stretch the bag gives it that extra oomph during the squeeze. So preload is actually to some advantage. Number 2 is contractility. That's based upon the calcium ion channels. So we'll see that in a moment. So contractility. If you've lost so much of your heart muscle, the myocardium, due to heart attack, then you may not have enough force to generate because of contractility issues. Number 3, this is your enemy. On top of your aortic valve, and that's the one I'm using as an example, on top of the aortic valve, we said that blood is going to regurgitate back when the heart is actually resting. Does that make sense? So blood's actually going to be pooling on top of the aortic valve when the heart is resting. Now, if that blood stays there, it's going to add weight to the other side of that valve. And so when the ventricle does get to squeeze, it's going to have to do what? Push harder. Let's imagine that you're at a McDonald's and you're trying to get into the restaurant, and everybody inside the restaurant is actually pushing on the door, and the door actually opens into the restaurant. And you're pushing on the door, which opens inward into the restaurant, and everyone inside is pushing back on the door. Are you going to be able to open that door? No. That's exactly what this afterload is like. So you do not want to have too much blood regurgitating on the other side, on the top side of your aortic valve. So afterload is actually your enemy. Because if you have a higher afterload, it means that the ventricle has to squeeze harder. Remember that McDonald's example. So if I somehow had to get into the restaurant and everyone is pushing on the door from the inside and the door opens inwards, I'm going to have to exert a lot of force. Okay? That's what it means when the heart has to work harder, and that's not a good thing. So let me back up real quick so you can write these down or make a note of them. Okay, we've talked about that one, and I'm now on this one. Okay? So this is your preload one. Affects the ability of muscle cells to produce tension. So preload was the one where you had fluid in your ventricle and it stretches the ventricle and allows more of a force, more of a push. Okay, next slide. Notice what it says. At rest, EDV is low because you really don't need it. Even your stroke volume is low at rest. You don't need too much blood being spit out into the blood vessel system because you're resting. And even the myocardium will stretch less. However, the moment you start exercising, what's going to happen? EDV will increase. More of your blood is going to be in there, in the heart, and it's going to stretch. And that stretching is going to allow more blood to come out. So at exercise, the stroke volume will go up. So at exercise, the stroke volume will go up. Okay? That's an important slide. So stroke volume will go up. Okay, next slide is the Frank-Starling law. Now, you don't know this, but you've already had that law by my saying. My saying says that what goes in must come out. So if the EDV in your ventricle increases, if more blood is now in the heart, then you're going to have to do what? Get more out. So your stroke volume will increase. If you have less, then you're going to get less out. So more comes in, more goes out. Less comes in, less goes out. So picture that from your ventricle point of view. If more is staying in the ventricle, less is going out. If more is coming in, more needs to go out. Okay, so that's how it works. EDV increases, more has to come out. Less comes in, less comes out. More comes in, more comes out. That's how it works. What goes in must come out. Okay, that's the rule. Now this paragraph right here says there's a limit to that. Okay, so there's a limit to that. The pericardial sac will allow the heart to only stretch so far. The myocardial tissue will only squeeze so much. So there is a limit to that. Okay? So again, the Frank-Starling law says what goes in must come out. More is coming in, more better leaving. Okay? And systolic volume, here's finally the definition, is the amount of blood that remains in the ventricle after your stroke volume. So this is the blood that remains in the ventricle after the stroke volume. So whatever you shove out and what's left over is your end systolic volume. Okay. This slide I've already shown, and now we have contractility. Now you already know that this is the common theme. What affects contractility? What affects the SA node? Your nervous system and your hormones. So electrical and chemical again. This slide's worth looking at. So make a note of that slide. This slide again is worth looking at. Remember I said it's always the parasympathetic that's going to reduce the contraction. So sympathetic will make it go faster and beat harder. So sympathetic, let me back up on that slide. So sympathetic makes it go faster and stronger. Parasympathetic, slower and not so strong. Hormones. Now, instead of me putting up this slide, I want you to read page 701. So all of page 701. Okay, that's an important page, page 701. Okay, so it goes with this slide, so page 701. Make sure you read that page. Afterload. This is something I said that is a bad thing. As afterload increases, stroke volume decreases. That's your McDonald's example. If more and more people inside the restaurant are pushing on that door, what's going to happen? You, being the stroke volume, you're not going to be able to do what? Go into that restaurant. Right? So less of you will go in to that restaurant if more is on that other side pushing the doorway closed. So higher the afterload, the less the stroke volume. This is a big deal as you get older because you're now going to have to do what? Generate more force so that you can counter this afterload. And that's a tough thing. Next slide. This picture is basically a summary of all that we have said so far. And this is found on page 700. So this is a summary of what we've said so far. The very last slide of the chapter is page 702. This picture is a summary. It is important. Now I'm going to go over starting this block right here. And we're going to add a little scenario to it. So let's say I'm a couch potato. If I'm a couch potato, what's going to happen? Skeletal muscle activity will drop. Okay? So I'll have less muscle activity, it will drop. That means I have less blood coming back to the heart. If I have less venous return coming back to the heart, that means I'm going to have less blood in the ventricle when the atria pump to the ventricle. So my preload will drop. If my preload drops, what will happen to my end diastolic volume? It will drop. If my end diastolic volume drops, my stroke volume will drop. That means I will have less blood coming out of the heart because I am a couch potato. In another example, we have this, blood volume. If for some reason I've injured my arm and I'm bleeding from my arm, what's going to happen? More volume is lost, which means I have less blood coming back to the heart, less of a preload, less of this, and then the stroke volume will also suffer. Okay? And so the heart's going to have to do what? Pump harder to get the blood to move around. Okay? So, same as down here. If the hormones don't work, then I'm going to have a problem with contractility. The last one is the blood vessels. If the blood vessels do not do a good job at removing the blood in the aorta away from the aortic valve, then I'm going to have a higher afterload. So if I can't get blood vessels to move the blood away from the aortic valve, higher afterload, which means I have a higher systolic volume. And a higher systolic volume means I'm going to have what when I do this? A larger number here, smaller number here would mean what? Less stroke volume. Let's do the math. If I had a number 2 here, so 2 minus 8. 2 minus 8 would give you a negative number, a smaller number. That means you'll have a low stroke volume. And we saw that on a previous slide. So higher the afterload, lower the stroke volume. So the heart's going to have to pump harder to get more blood out. Okay? So it's really important to have that scenario understood. All right, that ends it for chapter 20, and we're going to relate this to chapter 21.